Hey guys, today we're going to go over the Lophotrocozoans. So basically, I wanted to remind you all that your Species Spotlight project is going to be due next Sunday. So make sure to get that done. I've been thinking about extending the extra credit opportunity an extra week just so you can get that in before the next practical. But yeah, next Sunday your project is due. So please utilize office hours this week. If you have any questions whatsoever, just come through and come to office hours. You can either come on Wednesday or Thursday at 11 a.m. and the Zoom links are on Canvas. Okay. All right, so let's get started here. Oh my God, only 2 more labs left. We have ecdysozoans and echinoderms and chordates, and then practical 2, and we're done. Remember, practical 2 will open on November 16th and be available all week until the following Sunday. It's the same format as last time, so make sure you study accordingly. So let's do a little bit of review. We talked about fungi last week. We talked about ascomycota, which are going to be yeast, molds, morels, and truffles. Just a little bit about them. Remember, they have hyphae reproductive structures, ascospores, and that fruiting body, right, which is an ascocarp. Then we have basidiomycota. Basidiomycota are typical mushrooms that we eat, toadstools, rust, and smuts. Again, they have branched hyphae. And then finally, the lichens. It's not its own phylum, it's just another division, but it's between an ascomycete fungus, remember, and also a bacterium or some sort of photosynthetic component, right? So, or like an algae, so a bacterium or an algae. So those are examples of those. All right, so reviewing our sponges from last week. We have calcareous sponges, which are made out of calcium carbonate, right? Demospongae, which is made out of spongin and silica also. And then we have hexactinolita, which is just silica, okay? Our different cnidarians, we have the hydrozoans, right? Which example would be Portuguese manowar or hydras or lobelia colonies. We have the scyphozoa, right? Those are just our typical jellyfish that we see in the medusa form. Then we have cubozoa, which are our box jellies, also usually in medusa stage. And then anthozoa, which are our corals, sea anemones, and sea fans. All right, so I really like this phylogenetic tree. It's a SpongeBob tree, but it has just a lot of all the traits that you need to know on there. And they're all mapped out. You can just use this to study with. There are other examples online as well. All right, so like I said, last week we did cnidarians, porifera, and fungi. This week we're doing the Lophotrocozoans. So we're going to learn about protostomes and deuterostomes this week as well, and how to differentiate those two. So protostomes, protostomia, it means that the mouth is going to form before the anus. That blastopore is going to form into a mouth first. So this whole group here, these are our protostomes. So like I said, the blastopore forms into the mouth first and then the anus in the future. This also exhibits what's called spiral cleavage during embryonic development. It's literally cleaving and kind of twisting around as it's happening. So there are 2 divisions of protostomes. We have the Lophotrocozoans, which we're talking about this week. They grow by steadily increasing their body mass over time. So direct development, okay? This is called direct development. Whereas ecdysozoans grow by molting or ecdysis, okay? And they typically experience what's called metamorphosis, all right? And we'll cover those next week. So tissue layers, we have different germ layers, right? So previously in the cnidarians, we had 2 different tissue layers, meaning that they were diploblastic. We had the endoderm and the ectoderm. Now we're seeing the mesoderm, which is used to create things like muscles in the body. Okay, so before diploblastic, then we had triploblasts. You can be triploblastic with a not without a coelom, and then we have triploblastic with a coelom, which is like a true sac for organs and things. So we don't really quite have the organs yet. So like I said, triploblastic, divided by body cavity, which is that coelom, and then the coelom is just a space for your organs, right, lined with a mesoderm. So different types of coeloms that we'll see. We have an acoelomate, which is just no coelom at all. There's no body cavity, for example, platyhelminthes, which we'll talk about this week. Then we have pseudocoelomates, which is like a false coelom, pretty much. There is a body cavity present, but it's only partially lined with the mesoderm. And then the eucoelomates actually have a true coelom, and it's completely lined with mesoderm. For example, annelids or earthworms, okay? So before we were experiencing radial symmetry, now we're experiencing bilateral symmetry. So this is a major evolutionary advancement. We also experience directional movement or forward movement, meaning that we're moving in one direction, which just like it sounds, and also cephalization. Cephalization is a really important word that you need to know for your lecture exam as well as lab. But cephalization is just the idea that your sensory organs are kind of concentrated in one place. So think about your face being you're cephalized, right? So your eyes, nose, ears, and mouth, they're all kind of in one spot. Okay, so our first phylum that we're going to talk about is Platyhelminthes. These guys have a flattened body shape. They can be carnivorous or parasitic. They primarily live in marine environments. So these are all aquatic organisms, okay? And again, these guys are triploblastic, meaning they have all three tissues. They're acoelomates, they do not have a true coelom, and they are bilateral, meaning that you could cut them right down the middle and they'd be exactly symmetrical on either side. So really special advancements in the Platyhelminthes phylum. So they have a nervous system. They have two ventral nerve cords, which are basically a concentration of their sensory organs. And you can also call this a cerebral ganglion, right? It's a very primitive brain. They don't have a true brain, but they have this thing called a cerebral ganglion. They do have sensory organs. They're not image forming, so that's the closest thing to eyes that they're going to get. They have photosensitive eye spots right here. They do look like little eyes. They're kind of cute under the microscope. And then they also have those chemosensitive auricles, which are located in the back of the organism, not on this diagram. You'll see it in another diagram. And they do not have a complete digestive tract. So instead, they just have this pharynx here, which they kind of use to accept food and things. And as far as excretory, they have what are called flame cells. So that's how they get rid of waste and also osmoregulate water. So they're these specialized flagellated cells only in the Platyhelminthes, which we will see more pictures of. These guys also, as far as motility goes, they exhibit gliding motion, and they're also excreting mucus while they're doing that. As far as reproduction, they are hermaphroditic, so they have both male and female parts, which is great. They can even they can self-fertilize, but they have methods in order to prevent self-fertilization. So the sperm and eggs can develop at different times. So it's over time they're separated, so temporal separation. They do exhibit asexual reproduction. So what's really cool about these, which we would usually see in lab, is that we can actually cut these guys with a scalpel or a knife, and after a week of time, they will divide on their own. So you could cut them, you could cut them right down the middle if you want, just like they did here, and it would have two heads after a while. You could, cut them in half, And the top half would grow a bottom, and the bottom half would grow a top. Super weird. So yeah, they exhibit fragmentation. Very interesting stuff. So questions about these guys. If your lab has different, so you can your lab might have different text. Just follow what's on your lab. This is an older piece, but they generally have the same questions. So, how does the movement differ from the sponges and cnidarians? Could sponges even move? No, they're sessile, right? Cnidarians, how did they move around? What's the symmetry of this animal, the platyhelminthes? And is its movement related to its symmetry? Yeah, it is, right? It's bilateral, and it's also moving forward in forward direction. Observe its feeding. You can look up a video online. So these are not hyperlinked, so you can't watch them, but you can find videos online. And then prepared slide. We have pictures of those as well on the atlas online, or you can look up a photo. So to what anatomy did these germ layers give rise to? So ectoderm, what does that grow? Endoderm, what does that grow? And the mesoderm, what does that grow into? And then is this animal acoelomate, pseudocoelomate, or eucoelomate? Oop. All right, so tubularia is the next class that we're going to talk about. So so this is the class within Platyhelminthes, the phylum. These guys are free living, same thing, reproduce by sexual reproduction, but can also be fragmented, right? Trematodes are also in this group. So no cilia on the epidermis at all. They have suckers or hooks for adhesion. They can latch on to things. Most of these guys are going to be parasites. So they're very parasitic, even to humans, can definitely cause diseases. And again, a lot of these guys are hermaphroditic. So trematodes, define an endoparasite. What do you think endoparasite means? So parasite, endo would be on the inside, right? Parasite would be harmful. The next class within Platyhelminthes is Cestoda. These guys are also parasites. For example, tapeworms. They don't have any digestive organs, and they absorb food from the intestines of their host. So they have long, flat, ribbon bodies, and again, they have suckers and hooks for adhesion to their host. You can get these from poorly cooked meat or fish, so be careful when you're eating things. Make sure it's cooked through. So Cestoda, how do parasites within this class feed? I feel like you can answer that one. All right, so next phylum. So we are out of Platyhelminthes, moving into the next phylum called Mollusca. So Mollusca, these organisms live in marine environments. They can also be terrestrial, like snails, and they can also live in fresh water. Second largest phylum in the animal kingdom, very diverse. And these guys do have a coelom. It is reduced. They have an open circulatory system, and they experience sexual reproduction. So these are the groups within Mollusca that we're going to talk about. So these are the different classes, right? So we'll get to them in a moment. But most mollusks have 4 defining features, right? So these are the 4 features of a mollusk. They have a hard external shell. They have a thin mantle, which will secrete the dorsal body wall, which we'll talk about when we talk about the bivalves. They have a visceral mass. That's where the organs are located. And then a muscular foot for locomotion. Okay? So anatomy and morphology of these guys, their excretory system, they have what are called nephridia. That's how they excrete waste. Nervous system is an interesting arrangement of ganglia and a pair of ventral nerve cords. We also have an open circulatory system. So the heart has atria and a ventricle, and it floods hemocoel, which is the cavity for the heart, right? We also have a complete digestive tract. This is the first time we're seeing a complete digestive tract. And many of these guys have radula, which are little teeth. It's a chitinous tooth that they use to scrape food with. So advantages of these guys, they have the capacity to support a larger body, more precise control of movements. They can go in a forward direction, but they also are just a bit more advanced than the platyhelminthes. And they can also move faster, usually, than the platyhelminthes. So as far as reproduction goes, we have sexual reproduction this time. So we'll have male and female organisms, and they can also be hermaphroditic. So bivalves experience external fertilization. So they'll do broadcast spawning. So they'll put their eggs and sperm on the outside. So they become larvae before metamorphosizing into adults. So again, they are going to experience metamorphosis and not direct development, which is growing larger. Direct development is growing larger. Cephalopods, so these are things like the squid or the octopi, right? So they have separate sexes. The males produce a packet of sperm and excrete that into the female. Females take care of eggs and releasing them into the environment. They have no larval phase. So these guys are only growing bigger and experiencing direct development. Gastropoda, so this is our first class, gastropods. They're a very largest group within mollusca. They have that single spiraled shell, so these would include the snails, right? Secondarily asymmetrical as a result of torsion during development, so they experience torsion, which is that twisting movement. Larvae have bilateral symmetry, and they may have 1 to 2 pairs of tentacles. They also have statocysts on their heads because they don't have any eyes, usually. So that's just responsive to their environment, their spatial orientation. So which class within mollusca does not have radula? So it would be gastropoda. They do not have the radula. So class Polyplocophora, so these guys are flattened and they have 8 plates. Okay, so that's a question in your lab exercises. The mantle encircles the foot. We have about 6 to 80 pairs of gills, just depends on the organism, and they do not have any eyes or tentacles, and they have a very reduced head. Okay. So how many dorsal plates does this class exhibit? Remember, there are 8 plates. And how many eyes? They don't have any eyes, right? Class Bivalvia. So these are our bivalves. These are going to be our clams and oysters and things like that. And they're called bivalves because they have 2 valves that hinge, right? Right here. And they have, they have a posterior adductor muscle. Their head is reduced. They have a laterally compressed foot, in-current and ex-current siphons to siphon water, right? They respire through the mantle surface and or by the gills. Did it move fast? Sorry. What part of these animals does the name bivalve describe? Remember those 2 hinged valves, right? Okay, cephalopoda. So cephalopods, these are going to be our squid. So they have very well-developed heads with large image-forming eyes. The shell is on the inside and is reduced, even absent in some species. They do experience jet propulsion. That's how they move, okay, jet propulsion. They have a closed circulatory system. And they have chromatophores, which are those color-changing cells, right? Which can help them camouflage from predators, right? So these are incredibly intelligent animals. Like octopus are very, very smart, and they can even hunt prey and perform tasks. It's very interesting. So for these, I want you to draw the external morphology of the squid, okay? So I have pictures in this PowerPoint, but they're also on the digital manual. I want you to identify the internal anatomy and also draw the internal anatomy, label these things, okay? So this is what we would be doing in lab. We would dissect or look at the planarians, the platyhelminthes, and then we would also dissect the squid and look at all those organs. So this is that platyhelminthes slide that you needed to draw for question, I believe it's question 1. Yep. Question 1 in your lab exercises. Okay, so you can draw these things here. Gastropod slide. This is the snail radula that you'll need to draw for gastropoda. So it says observe and draw the snail radula whole mount. All right. Squid dissection. So here are all the labeled external anatomy. Okay. For the practical, you'll need to be able to identify these things. All right. So you could just have this up while you're taking the practical, I guess, but you'll need to be able to identify these parts. All right. So here's the internal anatomy, all labeled. Make sure to label those things that it says to in your lab exercises. And now we're going to get to annelids, so phylum Annelida. So these are our earthworms, polychaetes, and leeches. So annelid characteristics, these guys, obviously can be terrestrial, but they also can be marine or freshwater. They have a coelom. It's well developed too, so they're considered eucoelomates. So they have a true body cavity to put their organs in. They also have a complete digestive system. They have a mouth and an anus, as well as all of these other digestive organs: pharynx, esophagus, crop, gizzard, and intestine. Complete nervous system and sensory systems. So they have a cerebral ganglion, it's that little brain, and then a single ventral nerve cord, which you'll be able to see when we look at the inside. Excretory system, they have paired nephridia to excrete waste. Then they have a closed circulatory system, so blood vessel and multiple hearts, okay? Reproduction, these guys can be sexual or asexual. So when they're asexual, budding or dividing into two segments, you can cut them up. Many of these guys are hermaphroditic. So in earthworms, the mating pair will line up and exchange sperm, and then they shed mucousy tube, which is provided by the clitellum. So this little thick band right there, that's the clitellum. So that's where external fertilization will take place. The tube is going to dry out and then forms a cocoon, and then you have baby earthworms. So again, like I said, so before this is the class that you need to know. So Oligochaeta, this is our first class that we're talking about within Annelida, the group. Okay, you don't need to know this subgroup name Sedentaria. I need you to know the name Oligochaeta. So these guys are terrestrial. These are our earthworms. They have setae, which are hair-like structures that aid in locomotion. Okay, no parapodia. They don't have any little feet or anything like that. They feed on decayed matter using their gizzard. Segments are identical except for the clitellum. So the clitellum is that thick band right here where the arrow is pointing to. That's the clitellum that secretes the cocoon. So sedentaria, that's the name of that subgroup. It just means that they spend a lot of their time underground in burrows or tubes, okay? And they're also divided into this group because they have a clitellum. All right. So I want you to draw the earthworm dissection. So external and internal morphology, I think, is on the next question. So for this one, it says to draw the internal anatomy in your lab manual, but you don't have to. I just want you to draw the external one, mainly the clitellum, mouth, anus, and setae. Answer these questions. I just gave you the answers to a lot of these. If you can't find it, look it up. So is this animal triploblastic? Well, of course it is. It has muscles, right? So it has that mesoderm. And of course it has epidermis and other parts. So endo and ectoderm are also present. Next class we're talking about is Hirundo. So these guys are the leeches. This is our leech class. So predominantly fresh water. They don't have any feet, no parapodia, and they don't have any setae either. They have a flattened body with a reduced coelom. So they do have a coelom, but it's just very small. And they do have some mesodermal tissue in there. They have a clitellum when they're reproductively active, so it'll disappear otherwise. So do leeches have parapodia or setae? No, they do not, right? And our next class, I believe the last class, is Polychaeta. So these are polychaetes, usually are marine organisms, so mostly marine, very complex head and sensory appendages. These are very cool to look at, lots of pretty pictures. Paired appendages, so they do have parapodia with setae. They have little feet with the little hairs, the setae. Occasionally have a chitinous jaw. Most of these are going to be separate sexes, so they'll experience sexual reproduction more often than they would asexual reproduction. And then they're in this Aurantia subclass, meaning that they're free moving. They're not very sedentary, right? They're not in sedentaria. So I want you to draw the prepared slide of the Nereis with the parapodium. Look at the parapodium and the setae, okay? So here's our earthworm dissection. You would need to know these parts here. That black line is the ventral nerve cord. So yeah, just take a look at these parts. Make sure you know those. Annelida slides. So again, here's that the earthworm that you would need to draw, mouth, anus, clitellum, and whatever else it told you to draw. And then here's the parapodia with the setae. And then finally, you need to label all of these characteristics located on page 41 in your lab manual. Make sure you go back to page 41 and upload that to the Canvas page as well, okay? All right, so remember, next week, this lab is due. Also next week, your web page and your individual summary paper are due. And we'll also talk about ecdysozoans next week. So make sure you know what to do before next lab. And if you need anything, please come to office hours.